Hey there! Today, we're diving into the fascinating world of the brain, specifically how it works when it comes to attention, hyperactivity, and a few other quirks that make us all unique. So, buckle up as we explore the colorful neural landscape of ADHD and beyond in a fun and friendly way. Let's start with the superstar of attention, your prefrontal cortex. Think of it as your brain's CEO, helping you plan, organize, and keep your working memory sharp. But in ADHD, this CEO sometimes gets a bit distracted or overwhelmed. That's because the prefrontal cortex isn't working alone. It's part of a busy network that includes the dorsolateral prefrontal cortex, orbitofrontal cortex, and even the anterior cingulate cortex, which helps monitor your focus and emotional responses. Now, what fuels this network? Enter dopamine, the brain's feel-good messenger. It's like a reward system that motivates us and keeps us attentive. In ADHD, dopamine pathways, especially those involving dopaminergic neurons and receptors like D1 and D2, might not be firing on all cylinders. This can lead to difficulties with inhibitory control and sustaining attention. That's why medications like lisdexamfetamine dimesylate, a stimulant, help by increasing dopamine availability, boosting focus, and reducing impulsivity. But dopamine isn't the only player. Norepinephrine, another neurotransmitter, works alongside dopamine to sharpen your attention and alertness. Think of norepinephrine like a spotlight that helps you zero in on important stuff, especially in areas like the right inferior parietal and left ventrolateral prefrontal cortex. Some treatments target these systems as well, using reuptake inhibitors to keep these chemicals active longer. Behind the scenes, the brain's architecture is a complex dance involving structures like the ventral striatum and nucleus accumbens, key parts of the reward system, and the dorsal striatum, which helps with movement and habits. These areas connect through loops like the frontal orbital cortex striatum cerebellar loop, coordinating actions and emotional regulation. When these circuits are out of sync, it can lead to emotional dysregulation and impulsivity often seen in ADHD and even autism spectrum disorders. Neurotransmitters like glutamate and GABA also play crucial roles. Glutamate, via receptors like AMPA and NMDA, facilitates excitatory signaling needed for learning and memory, especially in regions like the hippocampus, which is vital for memory formation, and the entorhinal cortex, acting as a gateway to memory storage. GABA, on the other hand, provides inhibitory signals that help keep neural activity balanced. In ADHD, this balance can be a bit off, leading to hyperactivity or distractibility. Speaking of memory, the hippocampus is a key player. Think of it as your brain's librarian, organizing and storing memories. It's connected to the prefrontal cortex, forming a circuitry that's essential for cognitive control. The hippocampus has subregions like the dentate gyrus and cornu ammonis, which work together to process and retrieve memories. Interestingly, brain-derived neurotrophic factor, or BDNF, supports the growth and survival of neurons here, promoting synaptogenesis, the formation of new connections, crucial for learning and adaptation. Now, let's take a quick tour through the brain's emotional landscape. The amygdala is like your emotional alarm system, alerting you to threats or rewards, while the orbitofrontal cortex helps evaluate these signals, guiding decision-making and social behavior. In ADHD and related disorders, this system can be over or under-responsive, contributing to emotional dysregulation. Circling back to attention networks, the default mode network, think of it as your brain's idle mode, activates when you're daydreaming or mind-wandering. During focused tasks, the task-positive network takes over, involving areas like the lateral parietal gyri and the precuneus, helping you stay engaged. In ADHD, the balance between these networks can be disrupted, making it harder to sustain attention. Beyond neural circuits, there's also a rich tapestry of neurochemical processes involving thyroid hormones, the hypothalamic pituitary thyroid axis, and even redox homeostasis, which maintains cellular health. These factors influence neural development, like dendritic growth, synaptogenesis, and myelination. Think of myelin as the insulation on your brain's wiring, ensuring rapid signal transmission. And it's not just about chemistry. Our behaviors and skills are shaped by neuroplasticity, the brain's ability to change. Activities like mindfulness-based therapies, neurofeedback, and cognitive training can enhance neural efficiency, helping improve focus, emotional regulation, and self-control. Psychoeducation and group discussions also provide support, making the journey of managing ADHD a team effort. Finally, all these systems and processes are regulated within a broader context involving the limbic system, the visceromotor processing centers, and hormonal axes like the hypothalamic pituitary adrenal axis, your body's stress manager. Proper functioning here is key to maintaining emotional balance and overall well-being. So, whether it's a moment of focused attention, a burst of creativity, or a bit of impulsivity, your brain's intricate network of structures, chemicals, and pathways makes you uniquely you. And understanding these marvels helps us appreciate the complexity of neurodevelopmental conditions like ADHD and the amazing potential for growth and change within us all. Thanks for joining this whirlwind tour of the brain. Remember, no matter how busy your neural circuits may seem, there's always room for learning, growth, and a little bit of fun. Attention deficit/hyperactivity disorder, or ADHD, is often described with three familiar words: inattention, hyperactivity, and impulsivity. But the brain behind those three words is considerably more interesting and considerably more complicated. ADHD is not simply a matter of being distracted, restless, or occasionally making an impulsive decision. It involves large-scale networks responsible for attention, motivation, reward, executive functioning, emotional regulation, and behavioral control. In other words, the brain is trying to run the meeting, answer the emails, remember why it walked into the room, and resist the temptation to reorganize the entire refrigerator at the same time. At the center of this story is the prefrontal cortex, particularly regions such as the dorsolateral prefrontal cortex, medial prefrontal cortex, and orbitofrontal cortex. These areas contribute to planning, decision-making, working memory, behavioral regulation, and evaluating consequences. The anterior cingulate cortex also plays an important role. It helps monitor conflicts, detect errors, allocate attention, and regulate behavior. Together, these frontal regions form part of a larger cortical control system that communicates extensively with deeper brain structures. One of the most important partners is the striatum, which includes structures such as the caudate and putamen. Along with the globus pallidus and other components of the basal ganglia, these structures participate in movement, habit formation, motivation, action selection, and reward processing. Then there is the nucleus accumbens, particularly the ventral striatum, which is deeply involved in the brain's reward system. This is one reason ADHD research often focuses on motivation and reward. The brain does not merely ask, "Is this important?" It also asks, "Is this interesting enough to do right now?" That question brings us to dopamine. Dopamine is one of the major catecholamines involved in attention, motivation, reinforcement, and movement. Two commonly discussed dopamine receptors are the dopamine D1 and D2 receptors. Their activity contributes to how neural circuits respond to information and rewards. Dopamine-producing neurons originate in regions including the substantia nigra and the ventral tegmental area. The ventral tegmental area communicates extensively with the prefrontal cortex and nucleus accumbens, forming important components of the brain's reward and motivational circuitry. Another major catecholamine is norepinephrine, also called noradrenaline. Much of the brain's norepinephrine originates from the locus coeruleus. These neurons project broadly throughout the brain, including the frontal cortex, and influence alertness, attention, arousal, and the ability to respond to important information. So dopamine and norepinephrine are not simply chemical messengers floating around hoping someone will notice them. They are part of highly organized catecholaminergic systems that help regulate communication between different brain regions. And then serotonin joins the conversation. Serotonergic neurons are concentrated in the raphe nuclei, including the dorsal raphe nucleus. Serotonin signaling influences mood, emotional regulation, impulse control, cognition, and numerous other functions. Serotonergic receptors are distributed throughout the brain, allowing serotonin to influence many different circuits. The relationship between these systems is particularly important in corticostriatal limbic networks. These networks connect the frontal cortex with the striatum and limbic structures such as the amygdala. The amygdala contributes to emotional processing and salience, while the prefrontal cortex helps regulate responses to those emotional signals. In ADHD, researchers have therefore examined dopamine, norepinephrine, and serotonergic signaling, not as isolated chemicals, but as interacting systems. There are also other neurotransmitters involved. Glutamate is the brain's major excitatory neurotransmitter and plays an important role in learning, synaptic plasticity, and communication between neurons. GABA is the major inhibitory neurotransmitter and helps keep neural activity from becoming an uncontrolled neurological party. Glutamate signaling can involve NMDA receptors, while GABAergic signaling provides inhibitory regulation. Together, excitatory and inhibitory systems help determine how neural circuits process information. The brain's support cells matter too. Glial cells are not merely the maintenance crew hanging around while neurons take all the credit. They participate in signaling, metabolism, immune regulation, synaptic function, and the environment surrounding neurons. Research has also examined processes such as neurogenesis, neuronal differentiation, cell adhesion, and axon guidance. These processes help establish, maintain, and modify neural networks throughout development. And ADHD does not operate independently of the body's endocrine system. The hypothalamus communicates with the pituitary gland, also called the hypophysis, which communicates with endocrine glands throughout the body. Together, these systems help regulate homeostasis and physiological responses. One particularly important pathway is the hypothalamic pituitary adrenal, or HPA, axis. The hypothalamus releases corticotropin-releasing hormone. The pituitary responds by releasing adrenocorticotropic hormone, which stimulates the adrenal glands to produce cortisol. Cortisol then participates in a regulatory feedback loop that helps control the system. The hypothalamus also regulates growth hormone through growth hormone-releasing hormone and growth hormone-inhibiting hormone. And the hypothalamus-pituitary-thyroid axis regulates thyroid activity through signals involving thyrotrophin-releasing hormone and thyroid-stimulating hormone. Studies have also examined areas including the hippocampus, thalamus, anterior olfactory cortex, anterior cingulate gyrus, and nucleus accumbens. Some research has explored genetic variation as well, including polymorphisms associated with serotonergic neurotransmission and dopamine-related pathways. Findings involving particular alleles and repeat variations can be interesting, but genetics rarely provides a simple one-gene explanation for ADHD. The biology is considerably more complicated. Attention deficit/hyperactivity disorder, or ADHD, is often described as a problem with attention. ADHD involves systems responsible for attention, motivation, emotional regulation, learning, memory, movement, and executive control. And one surprisingly useful place to begin understanding those systems is mindfulness. Mindfulness is the practice of bringing attention to the present moment with non-judgmental awareness. The goal is to notice thoughts, emotions, sensations, and impulses before automatically reacting to them. This can support emotional awareness and self-regulation, particularly when emotional dysregulation makes feelings arrive with the volume turned up. Several brain regions participate in these processes. The anterior cingulate cortex contributes to attention, conflict monitoring, and cognitive control. The insula contributes to awareness of internal bodily states and emotional experiences. The prefrontal cortex helps with planning, decision-making, inhibitory control, cognitive flexibility, and other executive functions. Mindfulness-based stress reduction and mindfulness-based cognitive therapy use structured mindfulness practices to help people change their relationship with stress, thoughts, and emotions. Cognitive behavioral therapy, or CBT, adds cognitive behavioral techniques for identifying unhelpful thinking patterns and developing more effective behaviors. Dialectical behavior therapy, or DBT, takes this even further by emphasizing mindfulness, distress tolerance, emotion regulation, and interpersonal effectiveness. Acceptance and commitment therapy similarly emphasizes psychological flexibility, acceptance, and purposeful action. Emotional regulation can function like a feedback loop. Emotion intensity influences behavior, Behavior influences social interactions, and social interactions can feed back into emotional state. Learning to recognize that loop can make relationships, coping, and self-regulation more manageable. Neurotransmitters are endogenous chemical substances that allow neurons to communicate. They are released into the synaptic cleft and interact with receptors on other cells. Some receptors are ligand-gated ion channels, which directly influence ion movement across a membrane. Others are G-protein-coupled receptors, which activate G-proteins, effector proteins, and intracellular messenger systems. The cast of neurotransmitters includes glutamate, gamma-aminobutyric acid, or GABA, dopamine, serotonin, acetylcholine, norepinephrine, epinephrine, and several neuropeptides, including endorphins and oxytocin. Glutamate is a major excitatory neurotransmitter. GABA provides major inhibitory signaling. Dopamine contributes to motivation, reward, movement, and cognitive functions. Serotonin participates in emotional regulation and numerous other physiological processes. Norepinephrine contributes to arousal and attentional control, while acetylcholine plays important roles in attention, learning, memory, and sensory processing. Serotonin is produced primarily by neurons associated with the raphe nuclei in the brainstem. Tryptophan, an amino acid, is a biochemical precursor for serotonin. Serotonin transporters help regulate serotonin signaling by controlling its removal from the synaptic space. Dopaminergic neurons are concentrated in regions including the ventral tegmental area and substantia nigra. Their projections form several major pathways. The mesocortical pathway connects dopamine systems with the cortex and is associated with cognitive and executive functions. The mesolimbic pathway connects dopamine systems with limbic and reward-related structures, including the nucleus accumbens. The nigrostriatal pathway connects the substantia nigra with the striatum and is especially important for movement. And the tuberoinfundibular pathway connects hypothalamic dopamine signaling with the pituitary gland. Norepinephrine has another major pathway originating largely from the locus coeruleus. Its widespread projections influence attention, arousal, sensory processing, and cognitive function. Acetylcholine adds another layer. Cholinergic projections from the basal forebrain, including structures such as the nucleus basalis of Maynard, medial septal nucleus, and diagonal band of Broca, are involved in attention, learning, and memory. Cholinergic systems also interact with the hippocampus, cortex, thalamus, and striatum. The thalamus itself acts as a major relay and processing hub, participating in sensory gating and communication between different regions of the brain. The hippocampus contributes to learning and memory, while the cerebellum contributes not only to motor coordination but also to broader cognitive and regulatory functions. Together, the prefrontal cortex, cingulate cortex, hippocampus, amygdala, thalamus, striatum, cerebellum, and associated pathways form interconnected networks supporting planning, organization, problem-solving, working memory, decision-making, time management, and inhibitory control. The hypothalamic pituitary adrenal, or HPA, axis connects the brain's stress regulation systems with the endocrine system. Cortisol is one of the major hormones involved, and researchers have examined measures such as cortisol levels and the cortisol awakening response. Oxidative and nitrosative stress involve reactive molecular species that can affect cellular components. Researchers have investigated markers involving lipid peroxidation, antioxidant systems such as glutathione-related enzymes and catalase, inflammatory proteins such as C-reactive protein and interleukins, and compounds including malondialdehyde and nitrite and nitrate. Polyunsaturated fatty acids such as the omega-3 fatty acid docosahexaenoic acid and the omega-6 fatty acid arachidonic acid contribute to cellular membranes and signaling pathways. 1-carbon metabolism provides another biochemical network involving methionine, homocysteine, folate metabolism, methylation, and transsulfuration. Tryptophan can be metabolized through pathways that produce compounds including L-kynurenine, kynurenic acid, 3-hydroxykynurenine, quinolinic acid, and anthranilic acid. Other amino acids and precursors, including tyrosine, phenylalanine, histidine, glutamine, and L-cysteine, participate in different biochemical pathways. Tyrosine and phenylalanine are associated with catecholamine synthesis, while histidine is the precursor for histamine. Attention deficit/hyperactivity disorder, or ADHD, is usually introduced through attention, impulsivity, and hyperactivity. But underneath those familiar words is a remarkably complicated biological landscape involving sleep, neurotransmitters, metabolism, inflammation, synaptic plasticity, and even the gut. Sleep is one important piece of the puzzle. Sleep disorders can affect attention, cognition, emotional processing, and daytime functioning, which makes sleep particularly relevant when studying ADHD. Melatonin is one of the major biological signals involved in regulating sleep and circadian rhythms. Its metabolism can be assessed through compounds such as 6-hydroxymelatonin sulfate. Researchers have also investigated GABA, tryptophan, serotonin synthesis, and melatonin-related pathways when examining sleep and ADHD. Tryptophan is especially interesting because it sits at the intersection of several biological pathways. It can contribute to serotonin synthesis, but it can also enter the kynurenine pathway. And tryptophan metabolism doesn't happen exclusively in the brain. The intestine and its enormous microbial ecosystem play a major role. Gut microbiota can metabolize tryptophan into a variety of compounds, including indolamines and indole-derived metabolites such as indolacetic acid, indolapropionic acid, and tryptamine. Some of these metabolites are being investigated for their potential roles in intestinal barrier protection, antioxidant activity, immune signaling, and communication between the gut and nervous system. The intestine isn't simply a digestive tube with a very busy schedule. It contains a complex microbial ecosystem that participates in amino acid metabolism, fatty acid metabolism, and neurotransmitter-related biochemical pathways. Tryptophan can also be converted into kynurenine and downstream compounds such as kynureninic acid and other metabolites. These pathways intersect with oxidative stress, immune signaling, and neuroactive processes. Other metabolic compounds, including derivatives of phenylalanine, fatty acids, phospholipids, and catecholamine-related molecules, provide additional clues about how metabolism might interact with brain function. Our cells constantly perform chemical reactions that produce energy. Inside mitochondria, the electron transport chain helps generate ADP, the primary energy currency used by cells. But energy production comes with chemistry that can generate reactive oxygen species. These include superoxide radicals, hydrogen peroxide, hydroxyl radicals, hydroperoxyl radicals, peroxyl radicals, and singlet oxygen. Reactive nitrogen species add another layer, including nitric oxide, nitrogen dioxide, and peroxynitrite. The body has antioxidant defenses designed to keep these reactive molecules under control. Glutathione is particularly important, along with antioxidant enzymes and other molecules that help maintain redox homeostasis. Cells need controlled oxidation and reduction reactions for normal signaling and metabolism. Problems can arise when reactive species overwhelm protective systems or when redox regulation becomes disrupted. Researchers studying ADHD have therefore examined markers associated with oxidative stress and antioxidant defense, including glutathione, malondialdehyde, oxidized DNA markers such as 8-hydroxy-2, deoxyguanosine, vitamin-related antioxidants such as retinol and tocopherol, beta-carotene, and coenzyme Q10. Researchers have also examined inflammatory signaling molecules including interleukins, tumor necrosis factor, interferons, chemokines, and C-reactive protein. These molecules participate in immune communication and can influence cellular activation. Astrocytes and other brain cells also participate in immune and metabolic signaling. So the nervous system is not isolated from the body's broader immune and metabolic environment. Neurons communicate at specialized connections called synapses. These connections are not permanently fixed. They can become stronger or weaker, depending on activity. Dendritic spines are tiny structures extending from neuronal dendrites where many excitatory synapses occur. Their shape and structure can change in response to neural activity. Inside these tiny structures is an intricate molecular system involving calcium ions, glutamate receptors, signaling proteins, and the F-actin cytoskeleton. Glutamate can activate AMPA receptors and NMDA receptors. When appropriate receptors are activated, calcium can enter the neuron and trigger intracellular signaling cascades. Calcium can activate proteins such as calcium/calmodulin-dependent protein kinase 2, or CMKI, and protein kinase C. Other pathways involve calcineurin and changes in coughlin phosphorylation. One important example is long-term potentiation, or LTP, in which synaptic connections become persistently stronger following particular patterns of activity. Long-term depression, or LTD, represents another form of plasticity in which synaptic efficacy can decrease. A tiny change in calcium concentration can activate a molecular pathway. That pathway can change a protein. The protein can alter a synapse. And changes across many synapses can influence how neural circuits learn and process information. Dopamine also interacts with these systems. Endogenous dopamine can influence synaptic plasticity and hippocampal and cortical function, while dopamine pathways contribute to motivation, attention, and reinforcement. Medications such as clonidine and guanfacine act partly through alpha-2 adrenergic receptors and are used clinically in ADHD. Their effects illustrate another important principle. Regulating neurotransmitter signaling can influence neural circuits without simply turning them on or off. Mitochondria provide energy. Antioxidant systems regulate reactive molecules. Immune pathways communicate through cytokines. Gut microbes transform metabolites. Neurotransmitters regulate communication. Synapses change their strength. Dendritic spines remodel themselves. The brain is constantly balancing excitation and inhibition, energy production and protection, adaptation and stability. When researchers investigate ADHD, they therefore look at many levels at once: behavior, sleep, neurotransmitter regulation, metabolism, inflammation, oxidative and nitrosative stress, mitochondrial function, synaptic plasticity, and neural circuitry. The brain is a living network constantly adapting to sleep, stress, nutrition, hormones, neurotransmitters, immune signals, experience, and the environment. Mitochondria help cells produce energy, and neurons are particularly demanding customers. They need enormous amounts of energy to maintain electrical gradients, transport materials, release neurotransmitters, and keep synapses functioning. One of the central metabolic systems involved is the citric acid cycle, also called the Krebs cycle. It includes compounds such as citrate, aconitate, isocitrate, alpha-ketoglutarate, and oxaloacetate. Glucose can be broken down through glycolysis to produce pyruvate. Pyruvate can then enter mitochondrial metabolism, where it contributes to energy production and other biochemical pathways. And pyruvate has options. It can participate in reactions involving pyruvate carboxylase, or enter pathways involving aminotransferases and alpha-ketoglutarate dehydrogenase. Glutamate and glutamate dehydrogenase also connect amino acid metabolism with mitochondrial metabolism. So metabolism isn't just about producing energy. It provides the raw materials that cells use for neurotransmitter synthesis, biosynthesis, and maintaining cellular structure. Compounds such as oleamide and anandamide participate in signaling pathways involving lipid metabolism. Anandamide is an endocannabinoid, while oleamide is associated with biological processes including sleep and neural signaling. Fatty acid amide hydrolase, or FAH, helps break down certain fatty acid amides. Other molecules, including myristic acid, oleic acid, and arachidonic acid, participate in lipid metabolism and cellular signaling. Dopaminergic neurons are particularly energy-hungry because they have extensive projections throughout the brain. The nigrostriatal system, for example, connects dopaminergic neurons in the substantia nigra with the striatum and plays an important role in movement. Other dopamine systems contribute to motivation, cognition, and reward. The neocortex, hippocampal formation, amygdala, and cerebellum all communicate through complex networks, and researchers are interested in how metabolism and neurotransmission support these circuits. Neural communication also depends on the balance between excitation and inhibition. Glutamate provides major excitatory signaling, while GABA provides major inhibitory signaling. Experiments involving theta burst conditioning demonstrate just how dynamic these systems can be. Patterns of neural activity can change synaptic strength through mechanisms of plasticity. Inside cells, proteins called GT pathways act like molecular switches. They can alternate between GDP-bound and GTP-bound states, essentially changing between different signaling configurations. The RAS family includes HRAS, NRAS, and KRAS. These proteins interact with receptor tyrosine kinases and other signaling systems. When receptors are activated, signaling cascades can involve guanine nucleotide exchange factors, RAF proteins, and mitogen-activated protein kinases. 7-transmembrane receptors, also called 7TM receptors, can activate signaling through mechanisms involving G proteins and beta-arrestin. Receptor transactivation can further connect different signaling networks. Inside the cell, the endoplasmic reticulum and Golgi apparatus coordinate protein processing, lipid metabolism, and trafficking. A protein or membrane component produced in one location may need to travel considerable distances to reach an axon terminal or dendritic compartment. That brings us to another family of molecular switches: Rho, Rab, ARF, and RAN-GT pathways. These proteins help regulate cytoskeletal organization, vesicle trafficking, cell polarity, nuclear transport, and membrane dynamics. Rho signaling is particularly important for remodeling the cytoskeleton. And neurons are basically cytoskeleton enthusiasts. Their axons and dendrites depend on actin, microtubules, spectrin-associated structures, septin systems, dynein motors, and other proteins to maintain their architecture and transport materials. Dendritic formation, dendritic spine formation, dendritic maturation, and dendritic arborization all depend on carefully coordinated cellular signaling. The cerebellum provides another fascinating example. The cerebellar cortex contains specialized neurons including Purkinje cells, Golgi cells, stellate cells, basket cells, Ligaro cells, granular cells, and unipolar brush cells. Purkinje cells are especially interesting because their enormous dendritic trees receive extensive synaptic input. Maintaining that architecture requires continuous protein production, transport, energy, and cytoskeletal remodeling. Misfolded or damaged proteins can be processed through systems involving chaperone proteins, the proteasome, ubiquitin-dependent degradation, and autophagy. Macroautophagy and related processes help cells recycle unwanted components. Agrephagy specifically helps deal with aggregated proteins. Cells also have sophisticated transport systems. Anterograde transport moves materials toward axon terminals, while retrograde transport brings materials back toward the cell body. Dynein is one of the motor proteins involved in intracellular transport. Mitochondria can receive calcium signals from the endoplasmic reticulum, while the two organelles exchange information about metabolic demands. Proteins involved in ER mitochondrial tethering help maintain these connections. Voltage-dependent anion channels in the mitochondrial outer membrane also participate in communication and metabolite exchange. Redox regulatory proteins, including thioredoxin-related systems, provide another layer of cellular protection. Post-translational modifications such as palmitoylation can alter how proteins interact with membranes and other cellular structures. The dopamine transporter is another example of a protein connecting molecular biology with neurotransmission. It helps regulate dopamine levels by transporting dopamine back into presynaptic neurons. The blood-brain barrier carefully regulates what can move between the bloodstream and nervous tissue. Transport can involve systems such as clathrin-dependent endocytosis and other forms of vesicular trafficking. Proteins such as transferrin can participate in receptor-mediated transport, while molecules such as riboflavin and coenzyme Q10 are involved in broader cellular metabolism. Mitochondria are generating energy, proteins are being manufactured, molecular switches are changing states, vesicles are being transported, synapses are adapting, neurotransmitters are being released, and thousands of cellular processes are happening simultaneously. Attention deficit/hyperactivity disorder, or ADHD, is often described through three familiar features: inattention, impulsivity, and hyperactivity. But ADHD is a neurodevelopmental disorder involving a much larger biological network, including dopamine and serotonin signaling, metabolism, stress physiology, inflammation, and the neural circuits that connect them. One way to understand this complexity is to start with dopamine. Dopamine is involved in motivation, reward, movement, attention, and cognitive control. Dopaminergic signaling depends on several biochemical steps beginning with amino acids such as phenylalanine and tyrosine. Phenylalanine can be converted into tyrosine, and tyrosine can then be converted into L-dopa through the action of tyrosine hydroxylase. L-dopa is subsequently converted into dopamine. These reactions depend on cofactors and enzymes, including tetrahydrobiopterin, iron, and aromatic amino acid decarboxylase. Dopamine doesn't simply appear and then hang around forever. It can be metabolized by enzymes such as monoamine oxidase and catechol-methyltransferase. These pathways produce metabolites including 3,4-dihydroxyphenylacetic acid and homovanillic acid. And dopamine has another career option. It can be converted into norepinephrine, which can then contribute to epinephrine production. Enzymes including dopamine beta-related pathways and phenylethanolamine N-methyltransferase participate in this broader catecholamine system. Serotonin has its own supply chain. Tryptophan can be converted to 5-hydroxytryptophan, which can then be converted to serotonin. Different enzymes, including tryptophan hydroxylase 1 and tryptophan hydroxylase 2, participate in these pathways. Serotonin can subsequently be converted into N-acetylserotonin and eventually melatonin through enzymes including arylalkylamine N-acetyltransferase and N-acetylserotonin O-methyltransferase. Tryptophan can enter the kynurenine pathway. Tryptophan 2,3-dioxygenase and indolamine 2,3-dioxygenase can initiate this pathway, producing kynurenine. Kynurenine can then be converted into several downstream metabolites, including kynurenic acid, 3-hydroxykynurenine, 3-hydroxyanthranilic acid, quinolinic acid, and picolinic acid. Some of these metabolites interact with pathways involving glutamate and NMDA receptors, while others participate in processes related to cellular metabolism and energy production. The body is also constantly processing other amino acids, including glycine, serine, threonine, glutamine, glutamate, histidine, lysine, arginine, isoleucine, tyrosine, and phenylalanine. Modern metabolomics allows researchers to examine many of these molecules simultaneously. Instead of studying one chemical at a time, metabolomics looks at broad patterns of metabolites that may provide clues about biological systems. Researchers investigating ADHD have examined metabolites involving amino acid metabolism, catecholamine metabolism, serotonin metabolism, lipids, phospholipids, and sphingolipids. Lipids are particularly interesting because neuronal membranes depend heavily on them. Phospholipids form the basic structure of cellular membranes, while sphingolipids participate in membrane organization and signaling. Fatty acids such as linolenic acid, oleic acid, arachidonic acid, and docosahexaenoic acid contribute to membrane structure and signaling pathways. The balance between omega-6 and omega-3 fatty acids can influence the production of signaling molecules called eicosanoids, including prostaglandins, leukotrienes, and thromboxanes. Eicosapentaenoic acid and docosahexaenoic acid are important omega-3 fatty acids, while arachidonic acid is a major omega-6 fatty acid. Inflammatory signaling involves molecules such as cytokines, and researchers are examining how inflammation and neuroinflammation may relate to ADHD. Oxidative stress is another area of investigation because reactive molecules can influence cellular signaling, membranes, proteins, and other components of neural tissue. The hypothalamic pituitary adrenal axis, or HPA axis, coordinates important physiological responses to stress. The hypothalamus communicates with the pituitary gland, which influences adrenal hormone production. Cortisol is one of the major glucocorticoid hormones involved in this system. Cortisol acts through glucocorticoid receptors and participates in feedback regulation. This system influences metabolism, immune function, energy availability, and physiological responses to stress. Glucose homeostasis is another part of the picture. The brain requires a continuous energy supply, while hormones and metabolic pathways work together to maintain appropriate glucose levels. Even appetite can enter the discussion. Hyperphagia, or excessive eating, involves complex interactions among metabolic, hormonal, neural, and behavioral systems. It should not automatically be interpreted as a defining feature of ADHD, but studying appetite and metabolic regulation can provide another window into how brain and body systems interact. The biochemical pathways we've been discussing ultimately matter because they influence neurons and the networks those neurons create. Dopamine and serotonin signaling interact with neural circuits involved in attention, motivation, emotional processing, and cognitive control. Glutamate and GABA contribute to the balance between excitation and inhibition. Glutamine and glutamate are also connected through metabolic pathways that link neurotransmission with cellular metabolism. Researchers have also identified metabolites associated with catecholamine and serotonin pathways, including compounds such as norepinephrine and 3,4-dihydroxyphenylethylenoglycol, as well as serotonin-related metabolites such as 5-hydroxyindole compounds. Instead of one simple pathway, researchers are investigating an enormous network connecting amino acids, neurotransmitters, hormones, lipids, immune signals, energy metabolism, and neural circuits. It's a story about a developing nervous system operating within a biological environment shaped by neurotransmission, metabolism, stress regulation, immune signaling, and experience. Your brain is basically a very busy biological city. Neurons are constantly communicating through neurotransmission, while glial cells, immune cells, blood vessels, and the blood-brain barrier are all trying to keep the neighborhood functioning. And underneath all that activity is chemistry. Lipids are a major part of neuronal membranes. Cholesterol, phospholipids, triglycerides, and fatty acids help determine how flexible or rigid those membranes are. In other words, membrane composition influences fluidity, viscosity, permeability, and functionality. Cholesterol is particularly interesting because the brain needs it, but cholesterol also has to be carefully transported and regulated. Low-density lipoproteins, high-density lipoproteins, apolipoprotein B, cholesterol esters, and various transport mechanisms participate in the broader lipid economy of the body. And those lipids aren't just passive building materials. They can influence receptors, ion channels, enzymes, membrane proteins, and signaling pathways. That means lipid metabolism can potentially affect how neurons respond to neurotransmitters and how efficiently signals move from one neuron to another. Now, enter the fatty acids. Omega-3 and omega-6 polyunsaturated fatty acids are important components of biological membranes and can serve as precursors for signaling molecules called eicosanoids. Linoleic acid belongs to the omega-6 family, while alpha-linolenic acid belongs to the omega-3 family. Through enzymatic reactions, these fatty acids can be converted into molecules such as gamma-linolenic acid, dihomogamma-linolenic acid, arachidonic acid, and eventually other signaling compounds. Fatty acids are built from carbon atoms, hydrogen atoms, and a carboxyl group. Some contain double bonds, making them unsaturated. Those double bonds influence the physical properties of membranes. The body can then use certain fatty acids to produce eicosanoids, including prostaglandins, thromboxanes, leukotrienes, lipoxins, and other signaling molecules. Cyclooxygenase, lipoxygenase, and cytochrome P450 pathways help transform these fatty acids into different signaling compounds. Some prostaglandins and thromboxanes participate in inflammatory or prothrombotic processes, while other lipid-derived molecules can contribute to resolution of inflammation. That distinction becomes important because inflammation isn't simply an on or off switch. The immune system has to know when to respond, how strongly to respond, and when to stop responding. When regulation becomes disrupted, inflammatory cytokines such as interleukins and tumor necrosis factor can become part of a broader inflammatory environment. Microglia, the resident immune cells of the central nervous system, can become activated in response to various signals. Monocytes and other immune cells can also participate in systemic immune processes that interact with neurological function. The blood-brain barrier adds another layer of complexity. It is not merely a wall separating blood from brain. It is an active biological interface involving endothelial cells, transport mechanisms, signaling pathways, and immune regulation. When metabolic dysfunction, oxidative stress, or inflammation affects these systems, researchers become interested in how those changes might relate to neuroinflammation and neuronal function. Oxidative stress is another important piece. Mitochondria, those multifunctional organelles responsible for much of the cell's energy production, also regulate calcium storage, heat production, reactive oxygen species, and apoptotic signaling. Mitochondrial dysfunction can therefore affect cells in several ways at once. ADHD involves complex neurodevelopmental biology involving networks associated with executive function, inhibitory control, working memory, reward sensitivity, impulsivity, planning, and decision-making. Brain regions and networks often discussed in this context include the medial prefrontal cortex, orbitofrontal cortex, parietal regions, the default mode network, and frontal subcortical circuits. The hippocampus, amygdala, hypothalamus, neostriatal systems, and cortical regions also participate in broader networks governing cognition, emotion, motivation, and physiological regulation. At the cellular level, neurotransmission depends on an intricate choreography. A presynaptic neuron releases neurotransmitters into the synaptic cleft. Those molecules interact with receptors on the postsynaptic membrane. Ion channels open or close. Electrical activity changes, and intracellular signaling pathways respond. Calcium is especially important because calcium entry into nerve terminals helps trigger neurotransmitter release. Magnesium also has an important role in neuronal physiology, including its relationship with NMDA receptor ion channels. Fatty acids can also influence membrane receptors, ion channels, and enzymes, meaning lipid composition can interact with neurotransmission at multiple levels. Arachidonic acid, for example, can participate in signaling associated with neuronal activity, while lipid-derived prostaglandins and other eicosanoids can influence neurotransmitter systems. Finding differences in lipid metabolism, inflammatory markers, mitochondrial function, or neurotransmitter-related pathways does not mean that one abnormality single-handedly causes ADHD, autism, or another neurological condition. Genetics can influence metabolism. Metabolism can influence signaling. Signaling can influence inflammation. Inflammation can influence cellular function. And cellular function can influence neural networks. Even methylation can enter the story because epigenetic regulation can influence gene activity, including genes involved in cholesterol signaling and other metabolic pathways. Garlic, for example, contains compounds associated with the Alliaceae family, including alliin and S-allylcysteine sulfoxide. Enzymes such as alliinase can help generate sulfur-containing compounds. These compounds have been studied for antioxidant, anti-inflammatory, antimicrobial, immunomodulatory, and cardioprotective properties. Some sulfur-containing molecules can interact with reactive oxygen species and pathways involving NADPH oxidases, while glutathione provides another major component of the body's antioxidant defense system. So the next time someone says, "It's all in your head," remember that technically what's happening in the head involves an extraordinary network of chemistry, electricity, metabolism, immunity, and communication. Brain activity requires energy, and much of that energy comes from mitochondria. These microscopic powerhouses produce ADP through oxidative phosphorylation, using the electron transport chain to convert nutrients into usable cellular energy. Whenever cells use oxygen to produce energy, they can also generate reactive oxygen species, or ROS. These include molecules such as superoxide, hydrogen peroxide, and hydroxyl radicals. ROS can participate in cellular signaling and normal physiological processes. The problem arises when their production exceeds the body's antioxidant defenses. That imbalance is called oxidative stress. And the brain has some very good reasons to care about oxidative stress. Neurons have high energy demands, contain membranes rich in polyunsaturated fatty acids, and depend heavily on mitochondria. They also have limited ability to replace damaged cellular components compared with many other tissues. So when reactive molecules start accumulating, neurons can become particularly vulnerable. One major source of reactive oxygen species is the NADPH oxidase family, often abbreviated NOx. These enzymes sit in cellular membranes and transfer electrons from NADPH to oxygen, generating superoxide. Their structures contain transmembrane domains and cofactors, such as FAD, that help move electrons through the system. NADPH oxidases are molecular electron transfer machines. NOx activity can participate in normal signaling and immune defense, but excessive activation can contribute to oxidative stress and inflammation. Immune cells have their own impressive chemistry set. Neutrophils, for example, can use NADPH oxidase to generate reactive oxygen species as part of antimicrobial defense. Myeloperoxidase can then use hydrogen peroxide and chloride to produce hypochlorous acid, a powerful oxidizing molecule. Superoxide dismutase converts superoxide into hydrogen peroxide. Catalase can break hydrogen peroxide down into water and oxygen. Glutathione peroxidase provides another route for removing peroxides. Glutathione itself is one of the body's major intracellular antioxidant systems. When oxidative stress becomes excessive, polyunsaturated fatty acids in neuronal membranes can undergo lipid peroxidation. That process can generate reactive aldehydes, including compounds such as 4-hydroxynonenal and malondialdehyde. These molecules can interact with proteins, membranes, and other cellular components. Mitochondrial dysfunction can disrupt ADP production, increase reactive oxygen species, alter calcium handling, and interfere with the electron transport chain. Calcium is particularly important because neurons rely on tightly controlled calcium influx for communication and synaptic plasticity. Dopamine is essential for several neural functions, including motivation, reward, movement, and attention. Glutamate is the brain's major excitatory neurotransmitter and plays an important role in learning and memory. Processes involving glutamate contribute to long-term potentiation, synaptic plasticity, and memory consolidation. But excessive excitatory signaling can also contribute to calcium overload and oxidative stress. This means neurotransmission, metabolism, calcium signaling, and redox biology are not separate stories. Microglia are immune cells that reside in the central nervous system. Astrocytes also participate in maintaining the neural environment and responding to injury or stress. When inflammatory signals increase, microglia can become activated, astrocytes can undergo astrogliosis, and a broader glial immune response can develop. Cytokines, chemokines, interferons, interleukins, and tumor necrosis factor alpha can all participate in inflammatory signaling. Some cytokines promote inflammation, while others can have regulatory or anti-inflammatory effects. Oxidative stress and inflammation can also interact with the blood-brain barrier. The blood-brain barrier is maintained in large part by specialized endothelial cells and associated cellular structures that regulate what moves between the bloodstream and nervous tissue. Inflammatory signaling, oxidative damage, matrix metalloproteases, and changes in endothelial function can potentially alter this carefully regulated interface. Researchers therefore investigate relationships among oxidative stress, inflammation, immune signaling, and neurovascular function when studying neurological and neurodevelopmental conditions, including ADHD. Some studies have also examined compounds associated with antioxidant and anti-inflammatory responses. One fascinating example is allicin, a sulfur-containing compound associated with garlic. Allicin and related sulfur compounds have been studied for their interactions with cellular redox systems, including glutathione-related pathways. Other molecules derived from sulfur chemistry can participate in antioxidant responses and cellular defense mechanisms. One important regulator is nuclear factor erythroid 2, related factor 2, usually shortened to NRF2. NRF2 can influence antioxidant and cellular defense genes by interacting with antioxidant response elements in DNA. When activated, this system can increase expression of proteins involved in managing oxidative stress. Nitric oxide is a signaling molecule involved in vascular regulation, neurotransmission, and immune function. Nitric oxide synthase produces nitric oxide from L-arginine, with L-citrulline produced as a companion product. Different nitric oxide synthase isoforms are regulated in different ways. Some forms depend on calcium and calmodulin, while the availability of cofactors such as tetrahydrobiopterin can influence how the enzyme functions. When nitric oxide interacts with superoxide, it can form peroxynitrite, a reactive nitrogen species capable of contributing to nitrooxidative stress. Other systems can also influence this environment. Omega, 3 polyunsaturated fatty acids, for example, are involved in membrane biology and can influence signaling pathways associated with inflammation and cellular metabolism. Peroxisome proliferator activated receptor gamma, or PPAR gamma, is another regulatory pathway researchers study because it can influence metabolic and inflammatory processes. Other compounds, including hydrogen sulfide and various pharmacological agents, have also been investigated for their effects on oxidative and inflammatory pathways. Even brain-derived neurotrophic factor, or BDNF, enters the conversation because it supports neuronal survival, plasticity, and adaptive responses. Attention deficit/hyperactivity disorder, or ADHD, is usually introduced as a story about attention, impulsivity, and executive function. But underneath those familiar symptoms is a much larger biological conversation involving neurotransmitters, hormones, brain circuits, metabolism, and cellular signaling. One interesting concept researchers have explored is reward deficiency syndrome, the idea that altered reward processing may contribute to behaviors involving motivation, reinforcement, and impulse control. It is not a formal diagnosis for ADHD, but it provides one way of thinking about how reward circuitry can interact with behavior. And this is where dopamine enters the conversation. Dopaminergic neurons in areas such as the ventral tegmental area communicate with structures including the nucleus accumbens and prefrontal and limbic regions. These circuits participate in motivation, reward, learning, and behavioral regulation. But dopamine isn't working alone. Glutamate provides much of the brain's excitatory signaling, while GABA provides much of its inhibitory signaling. Dynorphin and other neuropeptides can also influence reward-related circuits. Now, let's introduce a hormone with a surprisingly large rhizome, glucagon-like peptide 1, or GLP-1. GLP-1 is an incretin hormone involved in glucose metabolism and insulin regulation. Drugs such as exenatide and liraglutide act on GLP-1 receptors, which belong to the large family of G-protein-coupled receptors. These receptors are molecular communication devices. When a signaling molecule activates a GPCR, it can trigger intracellular pathways involving cyclic AMP, or CAMP, calcium influx, protein kinase A, and CREB. The CAMPK-CREB pathway is particularly interesting because CREB can influence gene expression associated with cellular adaptation and neuronal function. So a signal that begins at a receptor on the outside of a cell can eventually influence what happens inside the nucleus. GLP-1 signaling is also connected to the brainstem. One important region is the nucleus tractus solitarius, or NTS, located in the brainstem. The NTS receives information from the body and participates in autonomic and metabolic regulation. The body needs to know what you've eaten, how much glucose is circulating, how much insulin is being released, and whether your digestive system should keep moving at full speed or take a brief coffee break. GLP-1 signaling helps coordinate some of those responses. Another incretin-related pathway involves GIP, or glucose-dependent insulinotropic polypeptide, which acts through the GIP receptor, or GIPR. GIPR is also a G-protein-coupled receptor, meaning it can activate intracellular signaling pathways involving CAMP and other molecular messengers. Researchers are interested in these pathways because GP and GLP-1 signaling may influence more than metabolism. Experimental research has examined possible effects on neuronal survival, neurogenesis, inflammation, and neural development. For example, signaling involving PK and CREB, along with pathways such as P38-MAPK and JNK, can influence cellular survival and stress responses. Researchers have also investigated relationships between incretin signaling and dopaminergic neuron survival. Now, let's travel from metabolism into developmental neuroscience. The brain develops through an extraordinary sequence of cell proliferation, migration, differentiation, and connection. The cerebellum, hippocampus, and other brain regions contain populations of developing neurons and glial cells that respond to a variety of molecular signals. In the hippocampus, researchers have studied progenitor cells and adult neurogenesis. The WNT signaling pathway is one of the molecular systems involved in cellular development and differentiation. GIP-related signaling has also been investigated in connection with WNT pathways, gene expression, and cellular regulation.